All right, everyone. So we are going to start different types of tissue now. And when we look at tissues, there are going to be four types: epithelium, connective, muscle, and bone. And of these four, epithelium, connective, muscle, and nerve. Look at that typo. Let's change bone to nerve. Okay. So here you'll see we have epithelium, connective, neural tissue, and muscle tissue. And of these four, epithelium and connective take a little bit more time to cover. There's a lot of categories under each, but neural and muscle is a lot easier. So you remember we've discussed this before, that when we talk about muscle, we said muscle comes in three flavors: skeletal, smooth, and cardiac. We know that skeletal is voluntarily controlled and it's striated. We know that cardiac is striated, but involuntary. And we know smooth is not striated, it is smooth, and it too is involuntary. So those are the three types of muscles, and we'll show you what they look like under a microscope. So check, right? Three that we need to know: skeletal, smooth, cardiac. Under neural tissue, you hear me say neural instead of nervous. I'm not a big fan of using the word nervous. I'm not a nervous person. I like the word neural. So the neural system, when we talk about the neural system, there are primarily two types of cells. There is the neuron and there are neuroglial cells. In lecture, you'll spend a little bit more time on the 6 different types of neuroglial cells. We're interested in the neuron, that is the electrically conductive or electrically excitable cell of the neural system. And it has 3 to 4, maybe 3 to 5 primary parts that you should know. And we'll go over those parts. Primarily dendrites, soma or cell body, and axon, and then maybe the myelin and myelin sheath, and maybe the axon terminal. All right, it's epithelium and connective that we have to spend a little bit more time covering. So the tissues have certain distinctive patterns, structures, and functions. And when cells come together, right, they make these different types of tissues. And when they have similar function, then those tissues come together and form different organs. So we're going to take a look at epithelium first. Now, epithelium deals with lining and covering things. It's a type of lining and covering type of tissue. It's going to cover the external surface of the body. Well, what do we call that layer of skin that covers the external surface of the body? We don't call it dermis. It's above that. Epi means above. It is epidermis. And FYI, just review the 5 different layers of the epidermis. We said that simple mnemonic was Cher loves getting skin Botox. Take the first letter of each. C for stratum corneum, loves lucidum, stratum lucidum. G, getting granulosum, stratum granulosum. Cher loves getting skin spinosum, stratum spinosum. And then Botox, stratum basal or germinatavum. Those are the 5 different layers. That's again in the epidermis. The dermis has the reticular and papillary layer. That's pretty much it. They line body cavities as well, and tubules. Tubules you will hear in the kidneys. There's a proximal convoluted tubule, a distal convoluted tubule. Tubules are tubes, right? I mean, the trachea is a tube. The esophagus is a tube. Your intestines are tubes. Those are larger, but they're still tubes. Blood vessels, arteries, veins, these are all tubes, and we have them throughout the body. Glands, these are specialized epithelium. And in terms of function, there's different types of epithelium that protect us against excessive friction, The skin, right, can protect us against bacterial invasion. The skin can protect us against chemical damage. But not just the skin, but even the tubule of the esophagus or even the stomach, that chamber or cavity of the stomach is designed in such a way that there's protection so that the acids within the stomach doesn't break down the actual stomach. Isn't it amazing that we have hydrochloric acid in the stomach, yet the stomach doesn't digest itself? So it's kind of neat how that's designed to protect. There's ciliated epithelium. Cilia, we've seen that when we covered the cell. The cilia or ciliated epithelium, we're going to find that in the respiratory tract, especially in the trachea. There are these ciliated epithelium, and the cilia are beating rhythmically from inferior to superior. And on the cilia, there's also mucus. And the mucus that sits on the cilia is produced by a particular type of cell called a goblet cell. And the goblet cell pushes out mucus, and it's going to stick to the top of the cilia so that if you breathe in any dust particles, the dust or the debris will stick to the cilia, the mucus on the cilia, and avoid making it to the deeper part of the lungs where the alveoli are. And because those cilia are beating from inferior to superior, it's a great defense mechanism of getting debris and foreign particles out of the lungs and up and out. Like in a cough or clearing the throat, like like that. It's enough to get things up and out. Epithelium is also used for absorption. There's special type of absorptive epithelium that line the stomach and the small intestines. And again, we absorb nutrients. We're absorbing water. We're absorbing vitamins and minerals. So epithelium is really important in terms of the GI tract. Think of the tract as tubules. So we're going to find epithelium there. Filtration. The kidney tubules, there's that word again. Kidney tubules, proximal and convoluted tubules, are lined with epithelium, and that can absorb, that can secrete, and that can filter blood. So the kidney is a major organ where a lot of blood is going through to absorb things, to filter things, to get things out of the body, and whatever the body doesn't want to get out to excrete, then we can reabsorb back in. So excretion, there's that word excretion, to eliminate, to get rid of. And secretion, glands. Glands can secrete their substances. Maybe it's secreting mucus. Maybe it's secreting saliva. Maybe it's secreting a digestive enzyme or a neurotransmitter or a hormone. That's secretion. And epithelium is also used for sensory reception. The epithelium that covers the body's surface contain many of the body's sensory organs to sense and feel. Here's the word cilia. Remember, ciliated epithelium. This is designed, these are long, slender extensions compared to microvilli, which are much smaller and thinner projections. The cilia is going to move secretions and fluids across the cell's surface. The microvilli is really designed to be like a sponge, to absorb things, to absorb nutrients primarily. Now, when we look at epithelium, we have to be able to look at it under a microscope, ask ourselves some questions about what we see in order to name what it is we're looking at. And one of the questions that's really important that you need to ask yourself is, number one, when I look at this at the apical surface, at the top layer, or the layer that's closest to the lumen, which is the whole of that tubule or vessel, does it look like there's one layer of cells, or does it look like it's multiple layers? So if we see that it's one layer thick, we use the word simple epithelia. If we see that it's more than one layer thick, we call it stratified. Okay, and I'll give you an example. I'm going to show you how we can pick this up in just a second when we look at a picture. The second question we have to ask is, what is the shape of these cells that we're looking at? Do they look flat and irregular shape? Then we're going to call it a squamous cell. Does it look like a cube or a box or a square? Then we're going to call it cuboidal. Do the cells that I'm looking at look like rectangles or buildings or columns? Then we'll call them columnar. So is it one layer? It's simple. If it's more than one, it's stratified. That's the first word of the epithelium. The second word of the epithelium is based on its shape. So we can have something called simple squamous epithelium, or simple cuboidal epithelium, or simple columnar epithelium. Or we can have it called stratified squamous epithelium, or stratified cuboidal epithelium, or stratified columnar epithelium. Now, I just want to skip ahead a second to show you what I mean. So here, when I'm looking at this, I can go, all right, this looks like it's one cell layer thick, and these cells are very flat and irregular. Now, when I look here, again, here's the bottom, here's the top. So the top is the apical surface. Here, it looks like there's 1, 2, 3, maybe 4 layers of these flat cells. So here, I'd call this simple squamous epithelium. But down here, I call this stratified squamous epithelium. We're not looking at the basement layer or what's down here at the bottom. We're looking at the top few layers. If I go to this one, this again is one cell layer. If I look at the one below it, it's multiple. Because these are not flat anymore, and these are not tall columns like these are to the right, but these are square or cube shaped, we're going to call this simple cuboidal epithelium. But down below, we're going to call this stratified cuboidal because it's more than one cell layer. It's 2 layers thick. Now, in terms of filtration or in terms of allowing things to diffuse, wouldn't you agree that if we wanted water or nutrients or gases like oxygen or carbon dioxide to pass through, it'll pass through this simple squamous layer pretty easily compared to something that's multiple layers? Yes, definitely. Diffusion of something from one side to the other, which means passing through it, will happen a lot easier if it was a simple squamous layer in relationship to stratified squamous. And the same thing is true if we're going to have water or certain nutrients to filter or absorb through something simple cuboidal compared to stratified. It would be easier to go through cuboidal that's simple compared to a stratified. Now let's look at simple columnar. These are columns. These are very tall rectangles or buildings. And notice how the nucleus, see how the nucleus here in the simple cuboidal, they're in the center. And then here in simple cuboidal, they're all aligned. The nuclei are pretty much in the center. So this is simple columnar. But if you look at the one below it, this is kind of tricky because look at the nuclei. They're at different levels. So at a quick glance, you'd be like, oh, it's stratified, right? I mean, simple, they're all aligned. So if we see them at multiple levels, it would have to be stratified columnar. But if you look closely, it's still only one layer thick. It's just one cell. But the nuclei are scattered at different levels. So it gives it this fake appearance that it's stratified. And that's why they use the word pseudo stratified columnar. Pseudo means false. So it's a false stratified layer and it's columnar. This one here, transitional. Transitional, think of a balloon, right? If you blow up a balloon, it stretches out, right? If you were to put, let's say, take a permanent marker and put like a line or a circle on the outside of the balloon, it may be very, very small, right? Like a small square. But once you blow it up and stretch it, it changes shape of whatever it is you just drew on the outside of the balloon. Transitional is the same way. So transitional cells are found on organs that can increase or decrease in size, such as the urinary bladder. If you don't have any urine in the bladder, it's small. But as the bladder is filling up with liquid, it stretches like the balloon, and we can see a variety of shapes. Some may look cube, some may look stretched and larger, so we call it transitional epithelium. Okay. All right. So let's go back now and continue with epithelium and say, okay, so based on the epithelium arrangement, depending on the number of layers, it could be simple epithelium or stratified epithelium. Okay. Then we look at the shape. If it's squamous, it's flat but irregular shaped. Primarily, squamous cells are used for protection, secretion, and diffusion. Whereas cuboidal, these are cube-shaped, these are found in the kidney tubules. These can secrete and absorb things that move across the gland or the tubular wall. So we're going to see cuboidal cells throughout the kidneys, especially the proximal and the distal convoluted tubules. You're going to hear that a lot when you get into anatomy and physiology too, when you learn the functional unit of the kidney called the nephron. And the nephron has filtrate that moves through the tubules. There's a proximal convoluted tubule and a distal convoluted tubule. And you're going to see cuboidal cells lining those tubules to help absorb and secrete certain materials. Then we have columnar. These are column shaped. The pseudo stratified we mentioned, they're simple columnar, but again, because the nuclei lie at different levels of the cell, it gives it more of a stratified appearance, but it's a false stratified. And oftentimes, wherever we have pseudo stratified epithelium, there's going to be cilia there. So we would call this pseudo stratified ciliated columnar epithelium. And I'll show you what that looks like in a picture. The transitional, remember, that's what I said is the kind of the balloon, like the urinary bladder, the ureters, parts of the urethra, things that can become larger and smaller. They kind of change their shape. So they transition. So we call it transitional epithelium. So again, simple epithelium is simple squamous, simple cuboidal, simple columnar. And then stratified squamous, stratified cuboidal, stratified columnar, and then those two little subspecialties. There's pseudostratified, ciliated columnar, and transitional epithelium. Okay, so those are the pictures that go along with it. And one of the things that I want to point out to you is something that I mentioned in a previous lecture, which is epithelium is a bunch of cells that are packed closely together with very, very little extracellular matrix. If you look at all of these are a bunch of cells that are closely packed together. Any one of these that you look at, and they're very closely packed with little extracellular matrix. I think in a previous lecture, I may have said, think of them as tiles placed nicely on the kitchen floor or something, right? How the tiles of the floor are closely packed together. And the grout between would be the extracellular matrix, and there's very little of it. Because connective tissue is just the opposite. Connective tissue is some cells with lots of extracellular matrix. So here with connective tissue, now we have a bunch of widely spaced cells that are separated by protein fibers and ground substance that we call the matrix, also known as ECM or extracellular matrix. Extra meaning outside of the cell. When you hear the word intra, it means inside of the cell. So if there's intracellular substances, that's inside the cell. Extracellular means it's outside of the cell. And connective tissue is the most abundant and the variable type of tissue that's found in the body. It's going to connect organs to each other. It can even connect to epithelium. We'll see that. It can give support and protection, like physical support and physical protection, as well as immune support and immune protection. And that's because of the blood, right? Blood is connective tissue. It is just considered liquid connective tissue. And here, when we talk about immune, yeah, we're talking about lymphatic fluid, but we're also talking about white blood cells. If you need to review the white blood cells, make sure you review the other lab lecture that I did talking about granular and agranular blood cells. The agranular was basophils, eosinophils, and neutrophils. And then the agranular is the monocyte and lymphocyte. Again, the three granular sites are the basophil, eosinophil, and neutrophil. And then the agranular means without the granules in the cytoplasm are the monocytes and the lymphocytes. Basophils, those produce histamine. When you're having an allergic reaction, your body's going to produce histamine. But the eosinophil tries to balance it out. It produces histamine ACE. So that's an enzyme that tries to break down histamine. And then the neutrophil, that's your primary responder, right? The neutrophil is high when there's a bacterial invasion. The lymphocytes go high when there's a viral infection. And then monocytes, those are always in circulation, and they find their way into a variety of tissues, but now they change their name from a monocyte into a macrophage. And depending on the type of tissue that that monocyte goes into, the monocyte can change its name. For example, if the monocyte is now in the liver, it's called a Kupffer cell. If it's in the lungs, it's called an alveolar dust cell. If it's in the brain, it's called a microglial cell. Okay. All right. Storage of energy and heat production. Well, adipose. If you look at the model of the skin and we see, okay, the top layer is the epidermis, then it moves into the dermis, and then there's the subcutaneous or the hypodermis. Way down at the bottom, there's a lot of yellow looking cells. That's the fat or the adipose or adipose sites. That's involved with heat production. But adipose also stores sugar. We call it a triglyceride. And fat cells also store toxins. They store toxins so that if you consume toxins, you don't want your liver to break them down too quickly. The liver may not be prepared to handle all those toxins at one time. So the fat cells release those toxins little bit at a time so that your liver can handle it. If you bombard your system with too many toxins, it'll throw your nervous system into shock. So we need to really protect the neural system. Little bit by little bit, your liver can detox these toxins. If you want to take a look at that, you can always look or research phase 1 and phase 2 of liver detoxification. You'll see that there are vital and very important nutrients and vitamins and antioxidants that the liver needs to support detoxification, which is why you will always hear doctors say, do not do a detox without doctor supervision, because people get very, very sick. The detoxification of toxins is more harmful and toxic than the initial exposure of the toxin if you're not properly equipped with the vital nutrients to handle the detox, right? So you get poisoned once by putting the toxin, and then your body's got to filter it out and clean it. And the byproducts of that could be very, very harmful to the body when you're trying to detox and break it down if you don't have the vital nutrients to support your liver. All right, movement and transport of blood, that's another important function. And now we're talking about red blood cells and the arteries, right? And how red blood cells and blood and white, how it moves and is transported through that highway of communication called your blood vessels. So now we're going to look at the different classifications of connective tissue. We have something called loose connective tissue. Now, if there's loose connective tissue, there's going to be dense connective tissue, right? Loose and dense, loose connective tissue and dense connective tissue. So let's look at loose connective tissue. There's three flavors that fall under that: areolar, which is loose connective tissue; adipose, that's your fat cell, that's loose connective; and reticular. So areolar connective tissue, that's going to provide the padding that separates your skin from the deeper layers, like from the muscle that lies deeper to it. And areolar connective tissue is made up of elastin and collagen protein fibers in the matrix. Then you have adipose, which are fat cells. We'll show you what that looks like. And the nucleus is off to the side of the cell with adipose, because a lot of students confuse adipose with a squamous cell. They look a little bit similar, but with fat cells, the nucleus is towards the periphery. With a squamous cell, the nucleus is always in the center. Then we have loose connective tissue, which is reticular, is also loose connective tissue. That provides the framework of support. So areolar, adipose, and reticular. We'll show you what those look like. Then we have dense connective tissue. Muscles, tendons, ligaments. These are dense, regular connective tissue. This is where you have collagen fibers that are very closely and tightly packed together. So it looks very, very dense and regular. But when you have these collagen fibers that are not closely packed in parallel direction and forms, but are in multiple direction, they're called irregular. So you have regular and irregular. Dense, irregular connective tissue would be like the skin, the dermis of the skin. It is made of collagen, but it's not parallel, tightly packed collagen fibers. They're in different directions to withstand force and friction and irritation that can come from a variety of directions. The direction of the fibers are really important, the way that they are laid down. If you've ever been massaged, your massage therapist doesn't just massage your back up and down, right? They can massage up, down, left, right in a circular direction because of the way the collagen fibers are lined. Now, there are people that have connective tissue disorders that have weak elastin and collagen, and their skin is very, very frail. Obviously, in those instances, there's a difference. But for most people, the dense, irregular connective tissue is irregular to withstand manipulation of the skin in a variety of directions. And then under dense connective tissue, there's elastic connective tissue. And we'll see elastic connective tissue in certain ligaments that give it their elasticity, such as the vertebral column, right? The vertebral column is very stable. The way the bones are aligned, they are very interlocked and stable. So the ligaments and connective tissue of the spine, you need to be very elastic so that you can bend and you can twist and you can rotate. So elasticity of the soft tissue around the spine is much more important than stability. You want more elasticity than stability around those vertebral joints. Now, if we're talking about the knee joint would be different. You'll have some elastic connective tissue around the knee because the knee does bend. It is very elastic, but there for the knee joint, it's much more important to have more collagen fibers for strength and stability of the knee, because the knee is the largest joint in the body, but it's also the most unstable joint in the body. Then you have fluid connective tissue, right? So fluid is what we covered: blood. There's the red blood cells, which are erythrocytes. They're going to carry blood gases like oxygen and carbon dioxide and nitric oxide, which will vasodilate the blood vessels, that nitric oxide. White blood cells, these are leukocytes. These are going to fight infection. And again, the granular sites are the basophils, eosinophils, and neutrophils. Those are granular in the cytoplasm. Remember, the basophil has that like very S-shaped nucleus. The eosinophil is bilobed, and the neutrophil has about 3 to 5 lobes to the nucleus. And then you have the monocyte and lymphocyte, which are agranular. The monocyte has that kidney bean-shaped nucleus without granules in the cytoplasm. And then the lymphocyte has the nucleus that's very round and almost occupies the internal space of the actual cell itself. And then you have platelets. Platelets help us form clots to help us stop bleeding. So if you get a cut, you're not going to bleed to death. You can clot. If you want to thin your blood, that's where baby aspirin comes into play, or fish oils and niacin and garlic and resveratrol or red wine, right? Those are all, or vitamin E. These are things that are known to thin the blood. Also, with fluid connective tissue, there's lymph. And lymph is the interstitial fluid. Now, that's not intracellular fluid. Interstitial is like extracellular. It's found between the cells on the outside of the cells. That is interstitial fluid. Then you have supporting connective tissue. That's cartilage. Now, cartilage tends to be avascular. Whenever you put the letter A in front of a word, it means without. So vascular is blood vessels, avascular is without blood vessels. So cartilage is avascular. And cartilage, we have three primary types: hyaline cartilage, elastic cartilage, and fibrocartilage. Hyaline cartilage is the one that we are going to see quite a bit in anatomy and physiology. You'll find it around your diarthrotic joints, which mean your freely movable joints, like the shoulder joint and the elbow joint and the wrist joint and the fingers and the hip and the knees and the ankles. These are all freely movable joints where bones come together. What separates the two bones, that's called a joint, and it's made up of articular cartilage. That articular cartilage between freely movable bones or joints is called hyaline cartilage, and it's the most common type of cartilage. Elastic, just like its name, it allows for a degree of flexibility. You'll find elastic cartilage in the ear and in the nose and in the eustachian tube, around the lungs, things that we need a degree of expansion. You can wiggle your nose or move your nose to the left and it bounces back. You can squish your ear and hold it in your palm and then let it go and it bounces back. That's elastic. Fibrocartilage, on the other hand, this is a very, very strong cartilage. It's a shock absorber. So we're going to have fibrocartilage in the knee. We call it a meniscus. That's fibrocartilage. We'll find it in between each vertebra. Those are called your intervertebral discs. And we'll find it where your pubic bones come together. There's a vertical disc there called a pubic symphysis. So pubic symphysis, intervertebral discs, and the meniscus is a fibrocartilage, very strong. Okay, and now we covered bone already. Bone is also falls under connective tissue. And remember, we talked about the osteon in another video. And some of the key words to be familiar with are osteocytes, concentric lamellae, the central canal or Haversian canal, osteoblasts. We said these are come from an immature bone, right? Meaning the osteoblasts produce matrix. It's immature, and then it becomes the mature osteocyte because it gets stuck in its lacunae. Okay, that's the cavity. Once the osteoblast gets stuck in its cavity of the lacuna, now it's called an osteocyte, which is a mature bone cell that helps to maintain bone. The osteoblast makes the matrix, the osteoclast dissolves and breaks the matrix down. So osteoblast builds up bone, the osteoclast breaks down the bone. The concentric lamellae are rings of that matrix that surround the Haversian canal or the central canal. Let me see if I can show you a picture of that. We talked about the interstitial lamellae, which are remnants of older osteons. The canaliculi are these extensions that come off of the osteocytes. And then let's see if I can show you. We did cover the endosteum and periosteum. The periosteum is the outer part of the bone. The endosteum is the inner part that's very close to the spongy bone. So these have the main parts that we spoke about. Make sure you review the parts of the osteon. The periosteum is the outer layer. The endosteum is the innermost layer. That's where you have the spongy bone. Compact bone is more towards the periphery, right? More around the outside. You could see the concentric lamellae here. You could see each of these osteons, lots of interstitial lamellae. You can see number 10s, all of the interstitial lamellae here and here. They're between each osteon, right? They don't make the full circle. So just review the osteon. We covered that in another video. Connective tissue, most of connective tissue is highly vascular, meaning it has good blood supply. The exceptions to that are going to be ligaments and cartilage. Ligaments and cartilage don't, and even tendons don't have the best blood supply. Muscles are pretty good, but tendons, not the best. So cartilage is going to get, and ligaments are going to get their blood supply and nutrition through diffusion. It's going to get it from all the adjacent structures that are next to it. It gets second dibs, okay? Now, because it doesn't have the best blood supply, whenever cartilage and ligaments are damaged, it takes a longer time for those tissues to heal. Okay, then we have three types of muscle. We said they're skeletal, cardiac, and smooth. Skeletal is voluntary, while cardiac and smooth is involuntary. Here, when you look at skeletal muscle, here it looks like it's showing the triceps muscle. It's very striated. You can see these striations, it means light and dark filaments. These are the nuclei, very striated. We can find these at any muscle that attaches to bone. Skeletal muscles have the ability to contract, which means to shorten and lengthen. When we cover muscles in more detail, we will go over the different types of contraction, like concentric, eccentric, and isometric. That comes when we cover the actual naming and identifying of muscles. Right now, we're still in the earlier stages of just understanding the difference between skeletal and cardiac. Cardiac is also striated. You do see light and dark filaments here, but here it is highly branched. You can see that there are many, many branches. So the description here, it's branched and it's striated. And you have these intercalated discs, which are these vertical linear bands going up and down. Those are intercalated discs. Those intercalated discs are not found in any other type of muscle except cardiac muscle. And it's where two different cardiomyocytes come together. If you were to separate them and look deep inside, you'll see the gap junctions. Remember, cells come together by different types of junctions to make tissues. They're tight junctions, gap junctions, desmosomes, hemidesmosomes, and adherent junctions. In this case, we're talking about gap junctions. Gap junctions are utilized in parts of the body where we have very fast, rapid forms of communication, such as muscle and nerves. Smooth, look at this. These look spindle shaped. They look very smooth, very spindle shaped with central nuclei. We do not see striations, and these are arranged in very close, tight sheaths. We find these in organs. They're involuntary. The uterus, the intestines, the trachea, arteries, these are going to be lined with smooth muscle. I have a good picture coming up showing smooth muscle of an artery. Now, arteries are really neat to look at because the lumen, what's close to the lumen, has squamous epithelium. But if we look deep to that, there's muscle because we know arteries can constrict and dilate. So it's a really, really cool slide to look at to see smooth muscle and epithelium. So you can look at connective tissue and epithelium in one microscopic slide. And then to finish up the four different types, we have the neural tissue. This is a neuron. If we look at a neuron has a few essential parts to it. First, you look at the cell body. That is called the soma, or the cell body. And of course, you see the nucleus in the center of it. And then we see lots of branches on the outside of that. Those are called dendrites. Dendrites comes from the Greek word dendra, which means branches or branches of a tree, highly branched. So information comes in to the cell body, coming into the dendrites. The dendrites are like receptors that receive input. So the dendrites take in information, the cell body processes that information, and then the electrical impulse goes away from the cell body through the axon. And notice the axon has some protection around it. Each of these circles is called a Schwann cell. S-C-H-W-A-N-N, Schwann cell. And the function of a Schwann cell, and that is one particular type of neuroglial cell. We have 6 types. One of them is a neuroglial cell or neuroglial cell. Its function is to produce myelin or myelin sheath. What myelin is nothing other than insulation or protection of this axon. And one of the functions of myelin is that it helps to speed up the action potential or the nerve impulse. It speeds up the conduction pathway of the electrical activity through the axon to get to the very, very end of the axon, called an axon terminal. And at the very end of the axon terminal are these swollen tips. The swollen tips of the end are called end bulbs. And guess what's stored in the end bulbs? In the end bulbs, we have something stored, a type of protein that is called a neurotransmitter. And the neurotransmitter is stored at the very, very end and can be released through a method called exocytosis. And that term should sound familiar because when we talked about the cell organelles, we showed you the Golgi apparatus. We showed you the endoplasmic reticulum, the ribosomes, the rough endoplasmic reticulum producing these proteins, and then it goes to the Golgi apparatus, which repackages some of those proteins. And one of those proteins can be shipped out from the Golgi complex in a vesicle that can move to the cell membrane. It can open up and release its contents. So that's what this can do. It can release its contents through a method called exocytosis. So those are the 3 neat parts of the neuron: the dendrites, the soma or cell body, and the axon. The axon is protected by the myelin sheath, which is nothing other than fat and cholesterol that is produced by the Schwann cell in the peripheral neural system. If we're talking about myelination of the central neural system, which is just the brain and the spinal cord, the oligodendrocytes do the same thing. So the oligodendrocyte and the Schwann cells are analogous to one another. They both produce myelin. It's just the Schwann cell does it in the periphery, in the peripheral neural system, whereas the oligodendrocyte does it in the central neural system, meaning the brain and the spinal cord. Okay, so let's escape through this. And now what I'd like to do is I'd like to show you the, let's see if I can I want to show you this one here, these histology slides. Whoops, let me go back. My bad. Let's go back in here. What I want to do is I want to show you the histology slides, and we're going to go through them and make sure that you can identify, and I'll walk you through and talk you through how to understand and ask yourself different types of questions. So you know what? I'm going to stop this video here and I'm going to create another one just for those slides. So let me stop the share and let's stop the recording here.